नो प्रॉब्लम नो प्रॉब्लम। प्रीव्यू तो आ रहा है उधर, कहीं तो प्रीव्यू ही नहीं आ रहा था। ओके ओके, गो लाइव, ठीक है। गो लाइव, ठीक है। और टेलीग्राम पे आ गया मैं क्या, टेलीग्राम टेलीग्राम लाइव हो गया हम। ओके ओके ओके, हेलो फ्रेंड्स, हेलो फ्रेंड्स। ओके, वेयर आर वी, नीचे। Okay, so we had sent a few questions for pre-test. This is just to give you an idea uh, what you'll be able to do at the end of the class. Okay, so I can see some of you have tried to attempt the questions. That's very good. So uh, once we have covered these topics, I'll present the same questions again to you to give you an idea of the gains that you have made after the class. Okay, so let's start with the class, and after the class we will revisit these questions again. We'll revisit these questions again, and we'll check your understanding. So as you know, today's topic is pH and acid base imbalance and buffers. So those are some of the things that we'll take a look. A lot of it you already know from the chemistry of 11th and 12th, but what is the medical aspect, the clinical aspect to your knowledge from chemistry to pH, buffers and acid base, we'll try to cover that part today here. All right, so a few things which obviously you already know, but I just want to highlight. First and foremost, what is pH? What is pH? I'm sure you already know. pH is the negative log. It is the negative log of H plus. Right? pH is the negative log of H plus, and we describe this pH in the range of 0 to 14. Obviously the values can be less than 0, more than 14, but in the lab those values are not very important. Most of the important values lie in the range of 0 to 14. In addition, the the pH value of 7 is said to be the neutral pH. Reason being, at this concentration, at this pH, the concentration of H plus and OH minus is equal. It is both of them. 10 to the power minus 7 is there. So this is the neutral pH. Now, normally in the chemistry lab, if the value of pH is less than 7, the solution is acidic, right? If the value of pH is less than 7, we call it acidic because the H plus concentration is more than what is seen in the neutral. Similarly, if the value is more than 7, we call it basic or alkaline. We call it basic or alkaline. In this case, the concentration of H plus is less than what is there in the neutral solution. So these are some values which you know, that in acidic condition the pH is less than 7, in basic or alkaline condition the pH is more than 7. This is what you already know from your chemistry. Okay, so like Sushil has asked you to confirm, I hope everyone is getting the clear audio and every one of you is getting the clear video. I hope nobody is having any issues with the transmission. All of you are able to hear me clearly and all of you are able to view us clearly. That is our hope. If you have any problem with transmission, please highlight it in our Telegram group so the IT team can take care of it. Okay, so moving ahead, these are the concentrations of H plus for the corresponding values of the pH. Obviously you already know this. For example, at pH 7 the concentration is 10 to the power minus 7 moles per liter. Okay, the H plus mass and mole will be similar. The molarity and mass will be same, because in this case mass of 1 gram will give us 1 mole of H plus. Okay, acids are H plus donor, this also you know, and bases are H plus acceptor, or they are hydroxyl group donors. They are hydroxyl group donors. Now, acids and bases can be of two types. Broadly, we can talk about acids and bases. They can be of two types. Some of them are strong. Some of them are strong. When we say strong acid or strong base, what is the important property? They will dissociate completely in solution. Complete dissociation occurs. It doesn't depend on the relative pH. It doesn't matter what is the pH. If we have a strong acid, it will completely dissociate. If we have a strong base, it will completely dissociate. It doesn't wait or get affected by the pH. On the other hand, we have what is called the weak acid and the weak base. Number one, they dissociate only partially in solution, and number two, whether they will dissociate at all or not also depends on the relative or the surrounding pH. It also depends on the pH of the surrounding. Okay, so this is about the strong acid and weak acid. Again, I'll say that you already know this. This is just a recap. This is the recap of information that will be requiring for this class. Okay, uh, so now we come to our body. What is happening in our body? What is the pH in our body? And how all the things come into play in our body. The pH is tightly controlled. When you go to the enzymes and proteins, that will see that all proteins and enzymes have an optimum pH at which they will provide the maximum activity. If there is a slight change in that pH, the enzyme activity goes haywire. So the body will try to maintain the pH very stringently in a very narrow range. And the most important point to note is extracellular fluid pH is 7.4. What do you mean by extracellular fluid? Mostly the blood. Obviously there are some extracellular fluid compartments besides the blood also, but we are currently focusing on the blood, meaning the pH of blood is 7.4. Normally in the physiological condition it is 7.4. Please note that we don't call it acidic pH, or we don't call it the alkaline or basic pH, although this value 7.4 is in the basic or alkaline range. We don't call it basic, rather we call it the body pH or the physiological pH. That range for this value is 7.35 to 7.45, and like I said, the body will very tightly regulate this pH. Okay, if there is gross fluctuation in the pH, the body cannot tolerate. And what is the limit of this fluctuation? If the pH falls below 6.8, or if it becomes more than 8, there is no possibility of survival for a human body and death will occur. All right, gross derangement starts occurring even when you shift maybe 1 or 2.1 or 0.2 or 0.3 units from the physiological pH, but at these values definitely the death will occur. If the value is less than 7.35, we call it acidosis, also known as acidemia. Value less than 7.35 will be acidosis or acidemia. Similarly, value more than 7.45 is alkalosis or alkalemia. These are the technical terms which you should be familiar when we talk about the acid base balance in the human body. The terms used are acidosis and alkalosis. These are commonly used. Sometimes the acidemia and alkalemia may also be used, but more commonly what we are using, the terms are acidosis and alkalosis. These are the common terms which are used to denote the derangement in the blood pH values. So diagrammatic representation to give you a pictorial memory. We said the normal value is 7.4, which is this one. Range 7.35 to 7.45. 7.35 to 7.45 is the range, and if you go below 7.35, as you can see here, this is the acidosis. If you go beyond 7.45, this is the alkalosis. Body can tolerate some amount of derangement, but beyond 6.8, death. Beyond 8, death. All right, so these are the ranges and the values you have to keep in mind. The normal range is 7.35 to 7.45. Decrease from 7.35 will be acidosis. Increase from 7.45 will be alkalosis. If the value crosses 8, death will occur. If the value goes below 6.8, again death will occur. Like I said, small changes in pH can produce major disturbances. That is why body takes great care to keep the pH in a very tight or the narrow range. Most enzymes function only with narrow pH ranges. Later on, when we are doing the enzymes, I'll show you there is the bell-shaped curve when we talk about enzyme activity. What this bell-shaped curve means, at the optimum pH there is maximum activity, but if you move from the optimum pH slightly, whether you go below or you go above from optimum pH, there's a sharp fall in the activity of the enzyme, very sharp fall in the activity of the enzyme. In addition, the acid-base balance also controls the movement of the electrolytes from one compartment to the other compartment. So if there is derangement of the acid-base values, in that case the electrolyte concentrations in the different compartment will also get affected. You know this from your physiology, that if there is any electrolyte imbalance in the compartments, it is going to have a lot of clinical presentations. You can have tetany, you can have neurological deficits, you can have arrhythmia. So a lot of changes can occur due to acid base imbalance, which again depends on pH of the different compartments. Okay, now the problem is the body produces more acid. The body produces more acid than, okay, the body produces more acid than the bases. So this is a big problem. What are the common sources from where we are getting the more acids? Majority of the food that you consume is acidic in nature. Majority of the food that you consume is acidic in nature, so foods are acidic. In addition, during metabolism, acids are produced. For example, for the metabolism of, let's say, carbohydrates occurring, you are metabolizing the glucose, anaerobic metabolism occurs. What you get? Lactic acid. All right, so a lot of acids are being produced in the body. And lastly, most importantly, no matter what substance is metabolized in the body, you know that the end product is going to be CO2, and CO2 itself gives rise to one of the important acids in the body, and that is the bicarbonate. So you can see continuously lot of acid is being produced in the body, and this acid is going to try and change the pH, which we had seen was in the alkaline range. It was in the value of 7.4, which we said is the alkaline range. It will try to affect that pH. This is just showing the example where most of the foods you can see. This is the cow milk, pH 6.6. Corn, we are very fond of that sweet boiled corn, pH 6. Cabbage, pH 5.3. Tomato juice, pH 4.2. Apple juice, pH 3. Gastric juice as well as a can of cola, Pepsi, Sprite, Thumbs Up, Coke, pH value 2. In the lab, try to take a pH paper strip in the range of 2 to take the gross pH paper, and you put it in a freshly opened bottle or can of your Pepsi, Cola, Thumbs Up, whichever which is the favorite cold drink that you take. Just put it and try to measure the pH. You'll be surprised to see the value coming out close to 2. So most of the food that we eat is acidic in nature, very few alkaline. For example, egg white, but how many of you consume raw egg white? No. Then some of the medicines, sodium bicarb, milk of magnesia, all of these are alkaline, but they're not common food stuffs. Most of the common food we have seen is likely to be acidic in nature. So the problem is how to control the pH, because most of our dietary activities, the metabolic activities, are going to bring that pH value under stress. Two ways. One is the buffer systems, and the buffer solutions, you already know, resist a change in pH. I'd like to add a small term here. They resist a small change in pH. The buffer systems resist a small change in pH. What is the composition of a buffer? Again, you know this, but we're just doing a recap. Buffers contain one of the two combinations. One of these two combinations will be there. Either we have a acid with its conjugate base and base with its conjugate acid. One of the two parameters will be strong. The second parameter, which will be complementary, will be weak. All right, you can have a weak acid with a strong base, you can have a weak base with a strong acid. All right, so we have alternating, or you can say complementary values of the acid and base. This is what we call a buffer. All right, you exchange a strong acid for a weak one, and as a result of the buffer, it will try to resist the change in pH. So if the buffer were not there, there can be a gross change of, let's say, pH value changes from 7.4 to immediately 6.4, but because of the buffering system, the change will be resisted, and rather than going way down to 6.4, the buffer value may just decrease by zero point 0.1 unit and stop at 7.3. This is how we talk about the buffer action. The buffering solutions will resist a small change in pH. They will try to resist the change in pH. In addition, we have some other mechanisms. See, this buffering system that I told you about, they are chemical buffers. They are chemical buffers. We'll see which are the chemical buffers which are available to help us to counter the change in pH. In addition to the chemical buffers, we also have what are the respiratory mechanism. Now respiratory mechanism is very important. It is mediated by exhalation of carbon dioxide. It is powerful, it can expel the acids, but it will work only with volatile acids. It will work with volatile acids, and it doesn't affect the acids which are there in the solution, which we call as fixed acids. Lactic acid concentration will not be affected by the respiratory mechanism. So how it is acting? The CO2 exhalation is controlled by the respiration. So CO2 will move out, and the concentrations correspondingly will be increased or decreased. So if you retain the CO2, if CO2 is retained, in that case what happens? The bicarbonate values will increase and the H plus concentration correspondingly increases. If on the other hand, you expel the bicarbonate, the H2CO3 values decrease, the H plus concentration correspondingly will decrease. So basically the respiratory mechanism affects the H plus concentration directly. H plus concentration, it is not able to change the fixed acids, but those volatile acids which can be converted to gases by the addition of H plus, those can quickly be exhaled with the help of the respiratory system. And lastly, we have the renal mechanism. A renal excretion, it has the ability to eliminate a large amount of acid. It can also excrete the base. The acid excretion occurs via H plus, and the base excretion via excretion of bicarbonate minus. Okay, so they can conserve as well as they can cause the production of the bicarbonate, and we'll see all this one by one. I'm just listing them how they are functioning, and they are one of the most important and effective regulator of pH. However, this renal excretion system is somewhat slow acting. It is somewhat slow acting. Remember, if there is kidney failure, there is likely to be failure of pH homeostasis, because the most effective regulation of pH is via the kidney. If the kidney is not working, you are very likely to see the pH imbalance in the patient. A quick look at rates of correction. If there is defect in if there is defect in pH imbalance is there, then what is happening, how they are acting. But because they are chemical in nature, they are able to quickly react and try to adjust the change in pH, so the chemical buffers will function almost instantaneously. All right, as long as the chemical buffers are acting, if they are active, you will not see any change. When do you see the change? You see the change when these chemical buffers get exhausted. When their buffering capacity is completely used up, only then you see the change in pH. Respiratory mechanism are so if we say this is fast acting, the respiratory mechanism is having the moderate rate of activity. It has the moderate rate of activity. It takes few minutes to hours to act. And the renal mechanism, I told you, is slow acting. It takes hours to days. The time duration required can vary from hours to days. So this is the rate of correction for the different mechanisms of buffering that we discussed: the chemical buffers, the respiratory mechanism, and the renal mechanism. These are the three primary buffering actions that we get to see. So like I said, first line of defense against the shift in pH are the chemical buffers. The important buffering systems are bicarbonate buffer system, by far the most important buffering system, the phosphate buffer system, and the protein buffer system. These are the three very important buffering systems in the blood. There are some other buffering systems, we'll see them one by one, which are not important in the blood, but we can see them in other compartments. The second line of the defense against pH shift are the physiological buffers, which are the respiratory mechanism and the renal mechanism. Remember, they are not so fast acting, they are somewhat slow acting, but nevertheless they are very very important, and they can correct very broad, or you can say gross, changes in the pH. Okay, so let's first come to the chemical buffers. At this point we are talking about the chemical buffers. Okay, like we said previously, the chemical buffers contain a relatively large concentration of an acid with its conjugate base, or a base with its conjugate acid. So strong acid, weak base, strong base, weak acid. So in this way we have the buffer composition. This is the buffer composition. The action of a buffer is described by what you call as the Henderson-Hasselbalch equation. The Henderson-Hasselbalch equation describes the action of the buffer. Here you have to note few things. What is pK? pK is what we call as the dissociation constant. You have derived the dissociation constant in your 11th and 12th in the chemistry section. So pK describes the dissociation constant. It is a fixed value for every solution. So we have the dissociation constant, and then we have the ratio of concentration of base and acid. Remember, this square bracket denotes concentration. The square bracket, I am sure you already know, it denotes the concentration. And the ratio of base upon acid, we take the log value, because if you recall, the pH itself is the log value. It was the negative log of H plus concentration. So here also, because to keep the values in the similar scale, you have to take the log values of the concentration ratios. So Henderson-Hasselbalch equation tells us that the pH in a system will typically be the pK plus log of concentration of base and acid. For example, if you take the bicarbonate buffer system, in this case the acid is the H2CO3 carbonic acid, and the base, the conjugate base, is the bicarbonate ion. So this is how we describe the Henderson-Hasselbalch equation, and we try to predict the pH at which it will be able to act. The pH at which it will be able to act. Now, two things to note here. Number one, at what pH the buffer is able to show a good action. So good action will occur in what range? In what range we'll see the good buffer? So remember, for good buffering action, the pH should be in the range of pK plus minus 1. For a good buffering action, the target pH where we want the buffer to act should be in the range of pK plus minus 1. How does it translate into the ratio of base and acid? If we say plus 1, this means the ratio is 10:1. If we say minus 1, this means the ratio is 1 upon 10. Alright, so the range in which a buffer will have a good action depends on the pK, typically for most of them, and 1 pH more than pK, 1 pH less than pK is the range in which the buffer will have the good action. So one parameter that we have to describe is the buffering action. Alright, second thing that you have to describe is the buffering capacity. What is buffering capacity? The amount of acid or base required to change the pH by 1 unit. And remember, the buffering capacity will depend on the concentration of absolute concentration of the acid base in the buffer. For example, if the concentration of acid and base is, let's say, 0.1 mole, then it will able to offer the buffering capacity equivalent to neutralization of this 0.1 mole of the chemical solution. On the other hand, if the concentration is 1 mole, it is able to provide more analyte to neutralize the substances which are trying to change the pH. And therefore, if you compare the two, obviously the buffering capacity will be 10 times in the second scenario. So capacity depends on concentration. Whether the buffering action will occur or not, it depends on the pH. So we have to know two things: whether a particular solution will be able to act as buffer, that's action. Okay, if it is able to act as buffer, whether it will be a good buffer or not, means does it have sufficient buffering capacity or not, that depends on concentration. So buffering action and buffering capacity, these are two different parameters which you have to keep in mind when we are talking about the chemical buffers. So now let's look at the chemical buffering systems. Most important by far for the whole body is the bicarbonate carbonic acid buffer system. The other buffering systems include the phosphate buffer system, the plasma protein buffer system, the hemoglobin buffer system. We'll look at them one by one. Okay. First, the most important buffering system, the bicarbonate or the carbonic acid buffer system. We already seen the Henderson-Hasselbalch equation for the bicarbonate carbonic acid buffer system, where the pH is equal to pK of log of bicarbonate. This is the base, and the acid is the carbonic acid. So we had seen the pH is equal to pK plus log of base upon acid. Sometimes in the Henderson-Hasselbalch equation, you may also see the symbol minus. Please note, from your 11th 12th we can change the symbol minus by exchanging the numerator and denominator. By exchanging the numerator and denominator we can change the symbol to minus also, but for the current scenario what we need is, okay, now few things that you have to know. Number one, what is the pK of the buffer system? What is the pK of the buffer system? In this case the pK is 6.1. The pK is 6.1. Now this is a very problematic value. We had said that the pH action, the buffering action will be good if pH is equal to pK plus minus 1. In this case the pK is 6.1, and if you add 1 to it, it becomes 7.1, right? It becomes 7.1, but the target that we need is 7.4. How is the bicarbonate carbonic acid buffer system able to act as a good buffer? Can anyone answer this question? Although the pK of the bicarbonate buffer system is unfavorable to act as a buffer in the blood, or for that matter any other compartment, important compartment, it is still the most important buffer system in the blood. How is it able to act as a good buffer in spite of this mismatch? It is a mismatch between the target pH and the pK of the buffer system. Anyone who can try and come to the solution, how to allow the bicarbonate buffer system to act as a buffer at the target pH 7.4 when the pKa is way far from the target pH. Anyone? Okay, Jeff says that bicarbonate buffer works only in alkalosis. Sandeep tells us quantity is a more. Very good. Anyone else? Anyone else? Remember, we had talked about two things. Okay, Kalp Manvar has given us a very interesting and correct answer. It has a high buffer capacity due to high concentration ratio 20:1. I'll come to that what it exactly means, but please recall what we are talking about. We're talking about two things. One was the buffering action, and the second is buffering capacity. Saravya Mantha has given a very clear cut statement. The ratio of acid and base is 20 is to 1. Priya, high concentration, it is somewhat correct answer. But remember, if you just say high concentration, it talks about capacity. It doesn't explain the action. It doesn't explain the action. It only talks about the capacity. Concentration describes the capacity. What about the action? How is it able to act as a buffer at this pH? So the ratio that has been mentioned by two of us, Kalp and Sravya, that contains the answer to our question. The ratio 20 is to 1. This is the physiological ratio of the concentration of the bicarbonate and the H2CO3. H2CO3, strictly speaking, gets translated in the partial pressure of a CO2. Okay, so the ratio is 20 is to 1 physiologically. If you solve this ratio 20 is to 1 and take the log, the value comes to 1.3. Now what happens? pK we said is 6.1. The value of this log is 1.3. So the target pH where it will have the best buffering action is 7.4. So bicarbonate buffer system is slightly unique from other buffering systems where we had said that the best buffering action comes in the range of pK plus minus 1. In case of the bicarbonate buffer system, because of the physiological concentration ratio, the best buffering action is seen at 7.4. Please note the ratio 20 is to 1, which is seen physiologically, is the deciding factor in this case, allowing the bicarbonate buffer system to act as the best buffer. It has the best buffering action. It has the best buffering action in the blood. There is a second aspect to this buffering action by the bicarbonate system, and that is because the lungs can readily dispose or retain the CO2, and the renal tubules can readily increase or decrease the rate of bicarbonate reclamation, meaning both the numerator and denominator can be easily changed. The degree to which we can carry out this change is, you can say, unlimited. The buffering capacity of the bicarbonate buffer system is theoretically unlimited. With the help of the respiratory and the renal mechanism, with the help of respiratory and renal mechanism, the bicarbonate buffer system has unlimited buffering capacity. So two things I told you: the bicarbonate buffer system has best action at pH 7.4, which is the physiological pH of blood. So it is the best buffer. It has the best buffering capacity because of the help it receives from the lungs and kidney, which can quickly change the numerator and denominator. Here we don't need to produce the carbonic acid or the bicarbonate ion, only we'll see later what is happening. Reclamation or excretion is occurring because these are automatically being produced in the body. Because of these two factors, best action and unlimited buffering capacity, the bicarbonate buffer system is the best buffering system in the body. So this is the dilemma that we had talked about. The pK is 6.1. So according to our calculation, plus minus 1, this should be the buffering range, but the target pH is here, and this is made possible because of the physiological concentration in the body. If you take out this buffer in the lab, in the chemical system, you will find that it is not able to act properly at the 7.4 pH, but in the body, because of the concentration ratios, it is the best buffer. I hope that part is clear about the bicarbonate buffer system. It is the most important buffer system. It has the best action at pH 7.4. It has unlimited capacity for action due to help from the kidney and the lungs. All right, let's go to the second most important chemical buffer. That is the phosphate buffer system. Now when we talk about phosphate buffer system, there is a very common mistake that a lot of us make. If you are not clear about it, we think about the phosphoric acid, right, and we think that this should be the acid, but please note that this is not so. The acid in the phosphate buffer system is not H3PO4. Rather, what you find is H2PO4 minus is the acid, and the conjugate base therefore is HPO4 2 minus. The very important point to note, what is the base and acid in the phosphate buffer system. Okay, that is first point. Second, what is the pKa for the phosphate buffer system, and that value is 6.8. Therefore the action range for this buffer system varies from 5.8 to 7.8. All right, the range of buffering action varies from 5.8 to 7.8. It obviously covers the physiological pH 7.4. all right, however, in the blood it has limited buffering action. If you remove all the bicarbonate buffer, of the remaining buffering action, only 5% is provided by the phosphate buffer system. So although the action can be there, the second part, capacity, is missing. Why? Because in the blood the concentration of the phosphate buffers is low. So the phosphate buffer system is a very important buffer system, but not in the blood, because in the blood the concentration which determines the capacity is low. The phosphate buffer system is important in the tubular fluids of the kidney. The concentration is very high. It is important in buffering intracellular fluid, again because inside the cells the concentration of the phosphates is significantly higher than the blood. So although good buffering action in the blood, but capacity is low, the buffering action is important in the tubular fluid in the kidney and the intracellular fluid. These are the two places where the phosphate buffer system will have the maximum action because of the concentration, simply because of the concentration. And then we go to the protein buffer system. We had seen that 5% was because of the phosphate buffer system. The remaining 95% of non-bicarbonate, the remaining 95% of the non-bicarbonate buffer system is albumin. It is because of the albumin. The reason for albumin being a very good buffer is the presence of histidine. We'll later see when we are doing the amino acids that histidine has a side chain of imidazole. It has a side chain of imidazole which has the pK of 7.3, which is closest to physiological pH. Please note, histidine is the only amino acid with buffering action. It is the only amino acid with buffering action because none of the other amino acids have the pKa close to physiological pH. Only histidine is able to provide the buffering action because its side chain has the pK close to the physiological value because of the imidazole side chain. Lastly, we get a little bit of buffering action from the hemoglobin. This is a bit of overlap from physiology, from the current scenario of the next and the instructions that we're getting from MCI. There is integration of the various subjects. So one integration is the horizontal integration, where we have to discuss overlapping systems from the different subjects. So we have a overlap with the physiology here, the hemoglobin buffer system. So I'll very quickly go through the hemoglobin buffer system. Obviously it will be covered again and in much greater detail in your physiology class, but just the part that we need to know here to understand the buffering provided by the hemoglobin. So this is the cell where metabolism is occurring, as a result of which CO2 is produced. You can see the CO2 is produced as a result of the metabolism, and this CO2 goes out into the plasma. The CO2 is coming out in the plasma. In the plasma, this CO2 can either get attached to some of the proteins which is there, albumin, or it gets dissolved due to the partial pressure. Some of it will get dissolved, but a large number of it combines with water, forms in the carbonic acid, and then dissociates into H plus, HCO3 minus. Some of it will attach to the protein. Some of the H plus will attach with the protein and form the HCO3 minus. Remember, in the plasma we don't have the enzyme carbonic anhydrase. So this reaction where CO2 combines with water to form the carbonic acid is a very slow reaction in the plasma. It is a very slow reaction in the plasma because carbonic anhydrase is not present in the plasma. So what happens to majority of the CO2? Majority of the CO2 actually enters inside the RBCs. Majority of the CO2 enters inside the RBCs, where it will combine with H2O in the presence of the enzyme carbonic anhydrase. Carbonic anhydrase speeds up the reaction by a factor of 10 to the power 8. It is the most efficient known catalyst. It increases the rate of reaction by power 10 to the power 8, 100 million times. The rate of reaction is increased 100 million times, so that quickly majority of the CO2 will combine with water and give us the carbonic acid, which dissociates immediately to form H plus and HCO3 minus. This HCO3 is exchanged with chloride. The SO3 will go out, the chloride will come in. The H plus which is formed, combines with hemoglobin, the oxygenated hemoglobin, and forms H hemoglobin, which results in the release of oxygen. This protonated hemoglobin is able to attach more of CO2. It is able to bind more of CO2. So number one, inside the RBC, the carbon dioxide is quickly converted to bicarbonate, and the protonated hemoglobin is able to bind more of CO2. So this is the dual buffering action of the hemoglobin. Although overall this buffering action is small, but the very important role of this buffering action is the production of bicarbonate in large amounts. Large amount of bicarbonate is getting produced. Okay, then you have the respiratory mechanism. I'll not go into the details of the respiratory mechanism because it depends to a great extent at the respiratory center in the brain. Again, I'll say this is the overlap with physiology, where you will study how the change in CO2 concentration and oxygen concentration derives the respiratory center, which in turns controls the amount of CO2 getting exhaled from the body. All right, so the CO2 concentration is able to regulate the respiratory rate of excretion. Second part is what is happening in the kidney. So we'll take a look of the various ways in which the buffering action is occurring in the kidney. three very important mechanisms in the kidney which have the buffering action. Bicarbonate, which is filtered at the renal glomerulus. See, this is the glomerulus, where from the blood vessels filtration is occurring. In this ultrafiltrate, most of the ions are filtered, particularly the bicarbonate ion is getting filtered along with the other anions and cations. All right, so the first step is reclamation of bicarbonate. This bicarbonate is reclaimed, and that is one buffering action. We can change the rate of reclamation. If you want more bicarbonate, rate of reclamation can be increased. If you want the bicarbonate to be lost, the rate of reclamation can be decreased. Hydrogen ion excretion, we'll see how the hydrogen excretion can occur. And third, production of ammonia. This ammonia buffers the hydrogen ion and forms the ammonium ion, which is then excreted. So these are the three mechanisms which act at the level of kidney and provide the additional buffering action. They provide the additional buffering action. Okay, first, how the reclamation of the bicarbonate occurs. So in the lumen, I told you that the bicarbonate has been filtered along with the other ions. All right, the bicarbonate, so this sodium which has been filtered is quickly taken up by the tubular cells, because sodium is a very important electrolyte. So sodium is taken up in exchange for H plus. All right, we have the sodium proton exchanger, antiporter. So it will take up the sodium and bring the H plus outside. This H plus will combine with the bicarbonate ion, form the carbonic acid, which will then quickly dissolve into CO2 and H2O. This CO2 moves inside. Now the equilibrium will always be in favor of the tubular cell for CO2 gradient, because inside the cell we have the carbonic anhydrase. And remember, the carbonic anhydrase will enhance the rate of reaction by 100 million times. It is one of the most efficient catalysts. So whatever CO2 is produced is quickly pulled inside the tubular cell, where under the influence of carbonic anhydrase, it is converted back to carbonic acid. The bicarbonate ion is produced. The H plus is used for exchanging another sodium. What is happening? There is a recirculation of H plus. H plus has been circulated, right? But the bicarbonate, which was taken up as CO2, is then transported into the interstitial fluid by sodium. So by this mechanism, the sodium and bicarbonate both have been absorbed. This is called the reclamation of bicarbonate. This is number one, first mechanism, reclamation of bicarbonate. I'll go through it quickly again. The sodium is moved in exchange for H plus. H plus combines with bicarbonate to produce CO2. CO2 moves inside the cell, where it is quickly converted back to the bicarbonate. This bicarbonate is pushed out of the cell along with sodium. The H plus thus produced goes back to the cell. So there is no net gain or loss of H plus. All right, no net gain or loss of H plus. But what is happening? The reclamation of bicarbonate is occurring. Second, we had said excretion of H plus. Excretion of H plus, and the most important buffer here we had previously said is the phosphate buffer. So H plus, which was moved out in exchange for sodium, it can quickly be taken up by the phosphate buffer, resulting in formation of H2PO4. Now this H2PO4 cannot be reabsorbed. It will combine with sodium and it will be excreted. Remember, the phosphates at the level of kidney are not reabsorbed. Typically they will be lost, so they will take up the H plus and they will result in excretion of the H plus. All right, third, we had said that ammonia production occurs. So ammonia production occurs with the help of two enzymes. One, glutaminase, and second, glutamate dehydrogenase. Classically, these two enzymes are part of the urea cycle, but we also see that they're playing a role in a buffering action at the renal tubules. Glutaminase will break the glutamine into glutamate plus ammonia, and the glutamate dehydrogenase, this is a reversible reaction which converts the glutamate to alpha ketoglutarate and produces the ammonia. Both this ammonia inside the cell gets converted to ammonia gas, and the gas is able to diffuse across the membrane. It is able to diffuse across the membrane. However, when it moves in the lumen, whatever H plus is there, it will quickly combine with the ammonium ion and it is lost in the urine. The ammonia will combine with H plus, form the ammonium ion, which will combine with any of the anions, and it is lost in the urine. So these are the three mechanisms by which the kidney is able to provide the buffering action: a reclamation of bicarbonate, excretion of H plus, and the production of ammonia. Okay, before we go to the acid base imbalances, I would request Dr. Sushil to repost question number 1 and 2 that we had posted previously. We had posted question number 1 and 2 in the pre-test. I would request Dr. Sushil to post the question number 1 and 2 again. You can try answering those two questions again. We had seen two questions in the beginning. Question number 1 was maximum buffering capacity of a buffer is at what pH? Maximum buffering capacity of a buffer is at what pH? So question 1 that we had asked was maximum buffering capacity of a buffer is at what pH? I'm sure you're able to answer this question now. If anyone is still not clear about this answer, I would like you to raise your hands. Okay, very good, very good. I'm getting some answers. Not yet, not yet. Okay, some of you had given the correct answer in the beginning as well. I would like to see how many if the number of correct answers have changed. Please try to give the answer to this question. Maximum buffering capacity of a buffer is at what pH? Less than pK, more than pK, equal to pK, has no relation. Okay, Jeff says equal to pK. Anyone else? Okay, Simrat, Jigar, answer again same, equal to pK, equal to pK. So remember we had said when the pH is pK will have the best buffering action, although the range of action can be pK plus minus 1. The range of action is pK plus minus 1. When pH is equal to pK, this is the value at which we will have the best action. Okay, Okay, and the second question that we had at this level, okay, I can see some options. D, I agree that it can be somewhat confusing that we had said buffering capacity depends on concentration, but in spite of concentration, the maximum buffering capacity for a given buffer will definitely depend on the pH. If it is outside the range of pH, then the buffering capacity doesn't come into play. Okay, second question that we had seen previously. Second question, why is the bicarbonate buffer system HCO3 minus H2CO3 is best buffer due to? This is the second question that we had asked. Combination of weak acid and weak base, pK is near physiological pH, it is good acceptor as well as donor of H plus, and last, its components can be increased or decreased in body as needed. Okay, now we are moving on to the second question. Second question, where we had said, we had talked about the best buffer being the bicarbonate buffer system. So why is it the best buffer system? Why is the bicarbonate buffer system the best buffer system? We have discussed the mechanism of the chemical buffers, so I would presume you are able to answer these two questions. Okay, D in second. Okay, Jeff says B or D. D, B. Okay, we have a very even spread of B, D. Some are saying B, some are saying D. Shravya says option B. Shreya says option D. Namrata says option D. Kalp says option D. Very good, very good. So this seems this is a very good question because we have an even spread of answers. Some are saying C, D, some are saying B. Please recall the pK of bicarbonate buffer system is 6.1. It is not close to the physiological pH. The pK of bicarbonate buffer system is 6.1. It is not, I'll repeat, it is not close to physiological pH. We had said in spite of pK being far from physiological pH, it is able to act as a good buffer. The answer was the ratios allow the bicarbonate buffer system to act as a good buffer. So obviously this option is out. Alright, you already know that we cannot have a combination of weak acid and weak base. Similarly, when I say good acceptor and donor, it means strong acid and strong base. Again, this is not a good buffer. This is not a buffer at all. So last we come, its components can be increased or decreased in body as needed. This is what we had highlighted again and again. Its concentration, the concentration of numerator and denominator both can be increased as per demand because of the respiratory and the renal system. Respiratory system will regulate the CO2 concentration. The renal system will regulate the concentration of bicarbonate. So the correct answer is D. Correct answer is D. Okay, very good, very good. So I can see a lot of people have got the point. Remember, the bicarbonate buffer system is best buffer in spite of its pKa being far from the physiological pH. It is in spite of, not because the pK is close to physiological pH. No, it is not that. Okay, let's go back and move on to the acid base imbalance. This was the background of the pH and the acid base homeostasis in the body, and here we'll move on to the acid base imbalance which may be seen in the body. Before you go to the acid base imbalance, few points which you should know and understand. Okay, acid base imbalance is a very important clinical aspect which you must know. uh, this is a very important foundation which you should clarify at this point, because all the patients that you will admit in your clinical setup, no matter which department, but more particularly important for where surgeries are being conducted, you will have to give some amount of fluid to the patient, or maybe the patient has come with some losses of fluids or some derangements of the electrolytes. So in almost all these patients you are going to see the acid base imbalance. So remember, acid base imbalances are one of the very important problems seen in the inpatient setting, and you should be very clear what you are looking at. Later on in the clinical setting, we'll tell you how to correct those acid base imbalances, but before that you should be able to determine what is the type of acid base imbalance that you're looking at. If you're not able to determine that, if you're not able to make the diagnosis, you cannot go to the management. So we can make that foundation here itself. Okay, we already said, if the pH is 7 less than 7.35, we will call the condition as acidosis, and if the pH is more than 7.45, we will call the condition as alkalosis. Okay, so acidosis and alkalosis is defined by the range 7.35 and 7.45. The body will try to correct the acid base imbalance. After the chemical buffers are exhausted, we'll see the derangement and the body will try to compensate, and this compensation is provided by the secondary systems. And which were the secondary systems? Respiratory and the renal system. Renal system is also known as the metabolic system. The renal system also known as the metabolic system. So sometimes, rarely, very rarely, if the derangement is very small, the compensation is able to bring the pH within the normal limits. In this case, we say that the compensation is complete, but this is rare. Remember, compensation is rarely complete. More commonly, what you see is the partial compensation. The body tries to correct the imbalance. It is able to correct it to some extent, but the range is still outside the normal value. So this is the partial compensation. More commonly, you will get to see the partial compensation in the clinical scenario. Complete compensation is rare. We get to see it very rarely. Okay, now here we talked about the acidosis and alkalosis. Depending on whether the cause for acidosis or alkalosis is respiratory or metabolic, they are further subdivided. Acidosis may be respiratory or metabolic. Alkalosis may be respiratory and metabolic. So we have overall four different types. Okay, we could talk about the compensation now. If the underlying problem is metabolic, in that case, the obviously the metabolic ability for correction has been used up, and primarily the respiratory compensation will come into play. Similarly, if the underlying problem is respiratory, metabolic compensation will come into play. See, the defect is occurring due to metabolic disturbance in first scenario. So obviously the compensation has to be provided by the respiratory system. Similarly, in the second scenario, defect is because of respiratory problems. So compensation has to be provided by the metabolic system. So the alternate system will provide for the compensation. The defect will cause the problem. The second alternate system will provide for the compensation. So what we will see in acidosis, principal effect of acidosis is depression of the CNS. All right, so you'll find what you find in these patients. You'll find generalized weakness, you'll find deranged CNS function, and the patient typically presents with disorientation, severe cases coma, and if you don't institute the management aggressively, the patient will die. So acidosis is a big problem. It has to be corrected on a war footing, and you have to manage these patients very aggressively. Alkalosis causes overexcitability of the central and peripheral nervous systems, causes numbness, it causes lightheadedness, it can cause nervousness, muscle spasm or tetany. This is one of the very important presentations. Convulsions, loss of consciousness, and death. Alkalosis less common, but again, if untreated, we have already said the acid base imbalances will cause death. If untreated, the acid base imbalances will definitely cause death. We had given the ratio ranges 6.8 if it is less than 6.8 or greater than 8, the patient will die. So what are the primary causes? What are the primary causes for acidosis and alkalosis? See, when we talk about acidosis, in that case there should be increased concentration of H plus. The H plus concentration can increase either due to accumulation of acid or when the bases have been lost. We'll see this when we are doing it later. All right, so either accumulation of acid or loss of bases. Similarly, if the pH rises, if you're talking about alkalosis, in that case there should be the decreased concentration of H plus. How it can happen? Either the bases are accumulating opposite, or there is loss of acid opposite of what was happening in acidosis. All right, so the change in amount of acid, the change in amount of base in the body, these are the two primary mechanisms which are causing the acidosis and alkalosis, which are causing acidosis and alkalosis. Okay, so like I said, the primary acid base disorders are metabolic acidosis, metabolic alkalosis, respiratory acidosis, and respiratory alkalosis. Let's look at them one by one. Metabolic acidosis. In case of metabolic acidosis, the bicarbonate concentration is less than normal. Okay, one more thing I want to tell you before we go to the acid base disorders. When we talk about acid base disorders, you will get three values. You'll get the value of pH. Remember, the normal value is 7.35 to 7.45, average 7. Then you get the value of PCO2. All right, 35 to 45 millimeter of Hg, normally 40. And then third value that you'll get is the bicarbonate concentration. The third value that you get is the bicarbonate concentration. Typically it will be 24, or rather we should say 22 to 26 milli equivalent per liter. Average value 24. These are the three values which you have to keep in mind. The three values which you have to keep in mind. Now, we already said what is acidosis and we already said what is alkalosis. In case of acidosis, there are two possibilities. What depending on the primary cause. One possibility is there is increase in PCO2. See what happens when PCO2 increases, the amount of carbonic acid increases and therefore the amount of H plus increases. So in acidosis, the H plus concentration increases, which can be because of increased PCO2. Alternately, there can be decrease in the bicarbonate concentration. There can be decrease in the bicarbonate concentration. So in this case, there is a relative increase in H plus concentration. H plus absolute amount is the same, but because the bicarbonate has decreased, there is a relative increase in the H plus concentration. So keep in mind these 2 points. In acidosis, either the PCO2 will be increased or the bicarbonate will be decreased. In alkalosis, we get the opposite picture. In alkalosis, we get the exactly opposite picture. Either the PCO2 decreases. If PCO2 decreases, the H plus concentration will decrease and it will cause the alkalosis, or you find there is increase in the bicarbonate concentration. Alright, bicarbonate concentration, when it increases, it will result in a relative decrease in H plus concentration. Right. Last point, if the change is in PCO2, obviously this is respiratory. If the change is in bicarbonate value, obviously this is metabolic. I'll repeat, the changes resulting in acidosis or alkalosis can be either due to change in PCO2 or due to change in the bicarbonate. If the primary cause is change in PCO2 value, respiratory. If the primary cause is change in bicarbonate value, it is metabolic. So remember, what are the changes that we'll get to see in the PCO2 values and the bicarbonate values in the corresponding acidosis and the alkalosis. This will help us to determine what is the primary cause. By looking at the concentration of the PCO2 and the bicarbonate, we will be able to tell what is the primary cause for acidosis and alkalosis. Once you understand that, only then you will be able to talk about compensation. Okay, so now let's talk about metabolic acidosis. When we say metabolic, obviously we'll talk about the bicarbonate concentration, and because we're talking about acidosis, we said in this case there will be decrease in bicarbonate resulting in a relative increase in H plus concentration. There is a relative increase in H plus concentration because the bicarbonate has decreased. Because the bicarbonate has decreased, what happened? Okay. Primary causes for metabolic acidosis. What are the primary causes for metabolic acidosis? More commonly, it is the production of acids. For example, ketoacidosis. We can have the lactic acidosis. There can be reduced excretion of acids, or there can be excessive loss of bicarbonate. Alright. An additional point to know here, in case there is production of acids in the body, if there is production of excess acid in the body, there will be an increase in a parameter called anion gap. This is a very important parameter. It helps us to distinguish between the different causes of metabolic acidosis. In addition, it helps us to identify disorders when the compensation is complete or when there is a mixed picture. So what exactly is anion gap? Anion gap is measured cations minus measured anions. Remember, the concentration of cation and anion in the blood will always be neutral. The blood is obviously always neutral. So if you measure all the cations and all the anions, the value will always be zero. But it is not possible. Physically, it is not possible or feasible to measure all the cations and all the anions. So what we do, we only measure the major cations and the major anions. So anion gap, most of the books will write it as concentration of sodium minus concentration minus the concentration of what's happening chloride plus bicarbonate. When you consider this formula, the value will be equal to 12 milliequivalent per liter. On the other hand, some books will give a slightly different parameter where along with sodium they also include potassium. In this case, the value would be slightly less and it is 8. If you include both sodium and potassium, the value will be 8 milliequivalent. But if you use only sodium, then the value is typically, sorry, it's the other way around. When you include sodium plus potassium, the difference is 12. I'm sorry for this confusion. And if you use only sodium, the value is 8 milliequivalent. Okay, so this comes into picture only when you talk about the metabolic acidosis. The anion gap will come into picture when we are talking about the metabolic acidosis. Okay, no, the previous values were correct. I'm sorry, the previous values were correct. Okay. Okay, what are the symptoms of metabolic acidosis? Symptoms of metabolic acidosis: headache, lethargy, nausea, vomiting, diarrhea, coma, and obviously death. But at the biochemical level, you should understand how the compensation for the metabolic acidosis will occur. The compensation will obviously be the respiratory compensation. It will be the respiratory compensation. Primarily what we see is increased ventilation. See what is happening in acidosis? We said the H plus concentration has increased. All right, one source of this H plus concentration is the bicarbonate, sorry, the carbonic acid, which comes from CO2. So if you are able to wash out the CO2, if you are able to wash out the CO2, the H plus concentration will decrease. The H plus concentration will decrease. So this is the respiratory mechanism. All right, the respiratory mechanism. By increasing the ventilation, what we are doing, we are trying to decrease the denominator. In acidosis, what has happened? H plus concentration has become more, so that the in metabolic acidosis the bicarbonate values have become less. So by decreasing the denominator we try to balance and bring the ratio to the previous value. About a diagrammatic representation, it will become more clear. See what has happened. Normally we have this balance. This is the CO2 and this is the bicarbonate ion. In metabolic acidosis there is loss of bicarbonate decreases, so we have this disbalance. So how do you, we cannot make more bicarbonate, so what we do, we try to decrease the H2CO3, and this is done by the respiratory system. It will wash out the CO2. At the same time some compensation is occurring by the kidney, but it is not enough, so we try to balance it. This is partial compensation. Complete correction occurs by management. We add more of the bicarbonate, so that the complete correction, the previous ratio 20:1 ratio is again achieved. So imbalance, loss of bicarbonate, the compensation, partial compensation by the two mechanisms. The kidney will try to reclaim more of bicarbonate, and the respiratory system will wash out the H plus. But complete correction only when you do the treatment, you provide the bicarbonate from outside. Lactate containing solution, ringer lactate can be given. So in that case we get the correct balance. So I hope this picture makes it more clear why we require increased ventilation is to decrease the denominator and restore the ratio between bicarbonate and the H plus concentration. Renal mechanism also acts, but it has limited role. Okay, then you got metabolic alkalosis. So in acidosis, the bicarbonate value was less than 22. In alkalosis, the value of bicarbonate is more than 26 milliequivalent per liter. Primary causes: excessive vomiting. In vomiting, what happens? The H plus from the stomach is excreted outside. Excessive use of alkaline drugs and certain diuretics, which will again wash off the H plus, Symptoms of metabolic alkalosis: slow, slow and shallow respiration, tetany. We had said this previously, and the electrolytes will often be depleted. Because electrolyte imbalance is there, we may also get the atrial tachycardia, or other dysrhythmias may be there. So how do we do the compensation? Obviously a respiratory compensation. We have to retain the CO2. What has happened in this case? The bicarbonate, the numerator has increased. To compensate, the denominator must also be increased, which is achieved by slow and shallow respiration. Renal compensative mechanism will be poor. I'll again show it diagrammatically what I am saying. What is happening? The bicarbonate has increased. Imbalance. Previously it was balanced, but now imbalance has occurred. So two mechanisms: either you try to decrease it, some action by the kidney; alternately you try to increase this value by the action of the lungs. Breathing is suppressed, so that more and more of CO2 is retained in the body. This is partial compensation. Ultimate correction, how? By providing the management from outside. When you start giving the chloride from outside in the body, the neutrality has to be maintained, so this chloride will start replacing the bicarbonate in the body, and bicarbonate excretion will start occurring. Okay, more and more of bicarbonate will be excreted, redistribution of the bicarbonate will occur, and the compensation will be complete by providing the management from outside. Respiratory acidosis, what we'll see in respiratory acidosis: any condition that decreases elimination of the carbon dioxide. Remember, when PCO2 is more than 40 millimeter of mercury, then we'll be talking about the respiratory mechanism being the cause. Two ways this can happen. The respiratory center in the CNS is depressed. This can be because of trauma, tumor, infection, or drugs acting at the CNS. Trauma in the CNS, tumor in the CNS, infection in the CNS, and drugs acting on the CNS. Alternately, the respiratory apparatus is affected. Chronic obstructive pulmonary disease, the fibrosis, acute respiratory distress syndrome, or trauma of lungs. Part of the lung is ruptured. Okay, so it is not able to carry out the respiration. So these are the causes which can cause a respiratory acidosis. Sign and symptom of respiratory acidosis. Some of this we have already seen. We are mentioning the clinical presentations: breathlessness, restlessness, lethargy and disorientation, tremors, convulsions, coma. Respiratory rate is rapid and gradually it will depress. Skin warm and flushed due to excessive vasodilatation, which is caused by CO2. This is a physiological aspect of the increase in CO2 values. So now how the compensation occurs. Buffer system will try to neutralize, but very quickly they will get exhausted. As long as they are able to act, the change in pH will not occur. Exhausted, only then acidosis occurs. At that time, the renal mechanism becomes the primary compensatory mechanism. The renal mechanism becomes the primary compensatory mechanism. Just to look at it diagrammatically, see what is happening. In this case, because the respiratory system is not working, either because of central effects or because of local effects, the CO2 retention is occurring. So this part has become super heavy, and what we can do, the body will try to increase the bicarbonate concentration, which is primarily done by the kidney, to also try to remove in the H plus. So the primary action by the kidney will try to increase the bicarbonate, but this will be partial compensation. Remember, I had told you the body is able to carry out only partial compensation. Complete correction will be done when you do the management. You're providing the lactate containing solution, ringer lactate is provided from outside. At that time, the complete rebalance will occur. And then we come to the most common acid base imbalance, the respiratory alkalosis. What is happening here? The carbonic acid deficit, PCO2 less than 35 millimeter of mercury. All right. Primary cause is hyperventilation. Luckily, most of the time it gets corrected by its own. Because of the change in the CO2 values, there is action at the central levels and it can get corrected of its own. So although it is the most common acid base imbalance, majority of it will not reach the clinical scenario. So conditions that stimulate the respiratory center, because of stimulation the excessive CO2 washout will occur. Oxygen deficiency at high altitude, pulmonary disease and congenital heart failure caused by hypoxia, acute anxiety. This is one of the very important causes for hyperventilation. One of the very important causes for hyperventilation is acute anxiety. In fever and anemia, the respiratory rate is increased. We see that respiratory rate is increased. Salicylate intoxication, cirrhosis, gram-negative sepsis, and one very important physiological scenario, severe exercise. Severe exercise also we find some degree of respiratory alkalosis. Compensation, buffer system will try to compensate. Once they are exhausted, then the renal mechanism will come into play. The three components we have already mentioned: decreased sodium H plus exchange, so that more and more of H plus will stay in the body. Decreased formation of ammonia, again more and more of H plus is retained, and we decrease the reclamation of bicarbonate, so that more and more of bicarbonate is lost. So what is happening here? The H plus is decreased, H2CO3 is decreased. So we try to decrease the bicarbonate by the action of the kidney. Ultimately, by providing management from outside the chloride solution, we are able to achieve the correct balance. All right. Remember, body correction will only result in partial compensation in all the images we have seen, whether it is the metabolic acidosis, alkalosis, respiratory acidosis, or alkalosis. The body correction is only resulting in a partial compensation. It is never complete. So, how do you diagnose the acid base imbalance? Here, you have to diagnose two things. Number one, what is the type of acid base imbalance? Acidosis, alkalosis, respiratory, metabolic. And second, whether the compensation is there or not. If it is there, what is the type of compensation? All right. So we had said the three parameters that we use: the pH, the CO2 level, and the bicarbonate level. The pH will tell you whether we are looking at acidosis or we are looking at alkalosis. So first look at the pH, so you know whether it is acidosis or alkalosis, and then look at the pCO2 and the bicarbonate values. You already know what should be the change in pCO2 in case of acidosis if it is respiratory, what should be the change in pCO2 in alkalosis if it is respiratory. Similarly, the change in bicarbonate in metabolic acidosis, change in bicarbonate in metabolic alkalosis. All right, this will help you to determine the primary cause. If a change in pCO2 corresponding to acidosis is seen, then it should be respiratory. Similarly, if the bicarbonate change corresponding to alkalosis is seen, then it should be metabolic. Okay, so like I said, if the cause is a change in pCO2, the problem is respiratory. If the cause is bicarbonate, then we label it as metabolic. Finally, look at the value which does not correspond to the change. There are two possibilities: either it is normal. If it is normal, there is no compensation. However, if it is outside the normal range in the opposite direction, then we can think of the compensation occurring. The change in the second parameter should be in the opposite direction. I'll show you with the help of a graph what I exactly mean by this. See, we look at the blood sample. The blood sample that we get is arterial blood sample, and the test is ABG analysis. ABG stands for arterial blood gas. This special way by which we take this sample. So we take a heparinized syringe, and we coat the heparin to the walls of the syringe, and immediately after taking out the sample we ensure that there is no contact with the air. So there are two ways: either you fold the needle or you put the tip of the needle in a rubber cap. So we have to ensure that there is no contact with the air, and we take the sample quickly for analysis. This is an emergency sample. ABG analysis is always an emergency sample. The result must be provided within 5 to 10 minutes. So by analyzing this arterial blood, we get result, and we look at the three parameters that I mentioned previously. First, pH. pH will tell us whether it is acidosis or alkalosis. Let's say the pH is less than 7.4. In that case, there are two possibilities. When we're talking about acidosis, there are two possibilities: either the acidosis is due to the bicarbonate values or it is due to the pCO2 values. If it is due to the bicarbonate values, meaning the concentration is less than 24 or rather 22 milliequivalent values, we say that it is metabolic acidosis. And then we look at the pCO2 value. If pCO2 value, instead of increasing in acidosis, the pCO2 value should be more than 40 millimeter of mercury. If it is decreasing, then we say that compensation is there. If there is no change, then no compensation. If it goes in the opposite direction, then we say that compensation is there. If we are talking about respiratory acidosis, the pCO2 value should be more than 40 millimeter of mercury, and if there is metabolic compensation, then the bicarbonate value should go in the opposite direction. Rather than decreasing, rather than decreasing, it should increase. It should be more than 24 milliequivalent per liter. The same thing can be done for alkalosis. Look at the bicarbonate for metabolic, and then for compensation look at PCO2 for respiratory, and then the compensation. So with this flow chart, you are very comfortably able to determine the primary cause and whether the compensation is there or not. There is a very useful tool in the clinical scenario to help you determine the type of acid base disorder. Again, you need the three values that I mentioned, the pH, PCO2 and the bicarbonate values, before you can make use of that tool, and that tool is known as the Davenport diagram. You can see it is a complex chart. It has three values. On the x-axis we have arterial blood pH. On the y-axis we have the bicarbonate concentration, and the remaining two axes are used to plot the PCO2 values. They're used to plot the PCO2 values. Now, using these three parameters from a patient sample, you can put the dot wherever you get. For example, we get a value of, let's say, the arterial blood pH is 7.2. All right, the bicarbonate value, let's say, is 18. All right, and the PCO2 value, let's say, is in the range of 40. Let's say it is in the range of 40. So your value will come somewhere here. 7.2, 18. So your value will come somewhere here. No, the 40 value is here. So that is a complex picture, unlikely. We'll have to take another values. So what we'll do, we'll revisit the questions that we had previously. We had I had posted three questions. Dr. Sushil, can you give us the three scenarios that we had posted in the beginning, and we can take a look at those questions. Now that we have discussed how to analyze the acid base imbalance, our friends should be able to solve those questions very easily. One question is there on the screen. You can try solving this. A patient is in intensive care because he suffered a severe myocardial infarction 3 days ago. The lab reports the following values from an arterial blood sample: pH 7.3, bicarbonate is 20 milliequivalent, normally it is 22 to 26, and PCO2 is 32 milliequivalent, normally it is 35 to 45. What should be the answer? You'll have to write the answer. I'm not giving the options. Your answer should have two parts. Your answer should have two parts. First, defect, whether it is acidosis, alkalosis, and which type, respiratory or metabolic, and whether compensation is there or not. If it is there, what is the type of compensation? I'm waiting for your answers. Okay, acidosis, what type of acidosis? Whether it is respiratory or metabolic. Not only defect, you have to also tell the type, whether it is respiratory or metabolic. Mm-hmm. Okay, so compensation, what type of compensation? Respiratory or metabolic compensation? Okay, Hena has given a very good answer. Metabolic acidosis and partial respiratory compensation. Compensation is also there. Very good, very good, very good, very good. So I'll just show you the answer. I'll show you the answer. The answer is metabolic acidosis with respiratory compensation. Very good, Jigar. That is the correct answer. That is the correct answer. Okay, so I hope at the end of this class today, you have been able to have a good overview of the acid base homeostasis in the body. What are the factors which are regulating the acid base balance in the body, the conditions in which the acid base imbalance can occur, and you should be able to confidently tell us the type of acid base imbalance and whether the compensation is there or not. At this point, I'll invite questions from you. If you have any doubts, any queries, any part you want me to repeat, please feel comfortable to list them. I'm waiting for queries and doubts from you. If you have any questions that you want to ask, you are most welcome. Any questions? I'll wait for doubts and questions from your side. Let me see if I have some more questions for you. Just hold on. Let's see if we have some more questions. Hmm, the drive will take up questions. Okay, let's see if we have some questions. Okay, I'm giving you more values. Please note pH 7.2, bicarbonate very low, 10 milliequivalent per liter, PCO2 30 millimeter of mercury. Okay, please try answering this. Okay, I can see some questions. Anion gap values. Okay, okay, I'll come back to anion gap. Sandeep, I'll come back to anion gap. Try answering this question. I'll come back to your doubts. I'll come back to your doubts. Very good. I can see people are asking doubts. Okay. Partially compensated metabolic acidosis. This means acidosis is there. See, if you see how we solve it, then the picture will become more clear. This means acidosis is there. For acidosis, there are two possibilities. Either there is increase in PCO2 or there is decrease in bicarbonate concentration. These can be the two causes in which we'll see the acidosis. Okay, what do you find in this scenario? Which condition is being fulfilled? We find that PCO2 is not decreased, sorry, not increased, rather it is decreased. So this condition is not fulfilled. Bicarbonate is decreased. So this is the primary cause. This is the primary cause. We said when we looking at bicarbonate, when you're looking at bicarbonate, it is metabolic. First we look at the pH value. From pH value we determine what is the type of disorder, whether it is acidosis or alkalosis. So pH value is less than 7.35, so it is definitely acidosis. Don't look at the values of bicarbonate and PCO2. If it is acidosis, there are two possibilities for acidosis. Either the PCO2 should be increased, number one, or the bicarbonate should be decreased. If PCO2 is increased, it would be respiratory. If bicarbonate is decreased, it will be metabolic. Now look at the two values. Is PCO2 decreased? Is PCO2 increased? No, it is not increased. PCO2 should have been increased, but it is not increased. So the respiratory is not the primary cause. Is bicarbonate decreased? Yes, the bicarbonate is decreased. Normal value is 22 to 26, and this is very, very low. So the primary cause is metabolic, right? Now we have determined that this is the primary cause. Again, look at the PCO2 value. PCO2 normal should be 40 millimeter of mercury. What is the value here? It is not increased. We know it is not increased. If it were normal, then we will say no compensation. But here it is not normal. It has gone in the opposite direction. Therefore, we say that respiratory compensation has occurred. We say that respiratory compensation has occurred. Okay, so metabolic acidosis with respiratory composition. Metabolic acidosis with respiratory composition. Now I'll try to answer some questions that we are seeing here. Number one, we take the arterial blood sample. First point, we take the arterial blood sample. First point. The test is called arterial blood gas analysis. The test is called arterial blood gas analysis. It gives us a large number of values, out of which we are very, very interested in three values. First, we want to know what is the pH. Second, we want to know what is the bicarbonate concentration. And third, we want to know what is the partial pressure of CO2. The result will also give you the values of sodium, chloride, and bicarbonate. It will also give you the values of sodium, chloride, and bicarbonate. Okay. So the pH will help us to determine defect. The values will help us to determine two things: primary cause and compensation. These values are generally not used, but if at all we make use of them, Then we calculate what is known as the anion gap, and the anion gap is increased when there is production of acids. Anion gap will be increased when there is production of acids. Okay, I'll see what all questions more are there. I hope the arterial blood sample test for acidosis alkalosis is clear. About anion gap, remember, normally no anion gap, meaning blood is neutral. Obviously it is neutral. It is neutral. Okay, but we only measure certain ions. We only measure certain ions. We have measured cations and we compare it with the measured anions. The measured cations, like I said, are sodium and potassium. The measured cations are chloride and bicarbonate. Obviously, they are not all the cations and anions. No, you cannot. As a result, when we measure, we get a difference. We get a difference. Alright, typically it is 12 milliequivalent per liter. Now, when there is more of unmeasured anions, let's say the values are 154 milliequivalent on both sides, same number of cations, same number of anions. And when you produce the acids, when there is production of acids, in that case, in addition to chloride and bicarbonate, we have more of anions. Alright, result is the portion of chloride and bicarbonate will decrease. The cations is the same, measured cations is the same, but the measured anions has decreased. In that case, what will happen to the anion gap? Anion gap will increase. Okay, okay. All right. So, what is the another question is ionized calcium increases in case of metabolic acidosis. See, I told you the changes in the pH. The changes in pH will result in changes of the electrolytes because of two reasons. They will affect how the ions are associated with the proteins, number one, and they will also affect the redistribution. They will affect the redistribution of the electrolytes from the cells into the blood and the vice versa, because the transporters are dependent on the pH. I hope that answers your question, Shravya. The ionized calcium increases in case of metabolic acidosis because the dissociation from albumin will occur when there is acidosis. Dissociation from albumin will occur when there is acidosis. Any more questions? Any more doubts? Or let's see, I'll give you one more question if we have it here. Okay. So remember, when you're talking about acidosis, sometimes the acidosis can be because of primary metabolic conditions, and this is one such condition which you should be able to answer. 55-year male, tachypnea means increased rate of respiration, and mental confusion. Blood glucose 350 milligram per deciliter. Normally it is in the range of 60 to 90. pH is 7. Urine shows ketone bodies. Nothing else is given in the question. Nothing else is given in the question, and you have to provide us with the diagnosis. Nothing else is given in the question, and the options are same: metabolic acidosis, respiratory acidosis, and respiratory alkalosis. What is the answer? Nothing else is given in the question. What is the answer? Everyone should be able to answer this question. It is a very simple and straightforward question. If you are having difficulty with this question, please ask me to clarify it. This is a very basic and fundamental question. Everyone should be able to answer this question, and you should be able to give the correct answer. Very good. Okay, so obviously what is happening, the acidosis is there, and because it is due to ketone bodies, it is metabolic in nature, it is metabolic acidosis. But can anyone give me the diagnosis? Sorry, can anyone give me the diagnosis? What is the this condition? What is the clinical condition that we're seeing here? Anyone who can tell me what is the clinical condition that we're seeing here? What is the clinical condition that we are seeing here? This is diabetic ketoacidosis. So like that, there can be certain scenarios where there is excess production of acid in the body, and in those cases there can be derangement in the anion gap. Very good. Kalp has given the answer: diabetic ketoacidosis with metabolic acidosis. Very good. So I hope it all of you are comfortable now with very good, you're comfortable now with the acid base homeostasis and the acid base related disorders. You should be able to very comfortably and confidently able to diagnose the conditions, at least the primary condition, and whether or not the compensation is available in these scenarios. If you're able to do that, I will think my today's job is done. And thank you all for attending this session. Our next class will be on 18th as per the schedule that I have received, and on 18th we'll talk about enzymes. We'll see what you should be knowing about enzymes. We'll take a look at the enzymes. Thank you very much. It was great fun being with you, and thank you for all the interaction and that you've had. If you have any doubts, if you have any queries, feel free to send them across on the Telegram group. The Telegram group is open to you for sending your doubts and queries. And if there are no more questions, I would love you to just give me a thumbs up so that I know that you are comfortable with today's discussion. Thank you very much. इसको बंद करोगे? हाँ, टीम में नहीं है, होगा टीम में, ऑन होगा देखो। 523 AIR